Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 1 for May 2023. Let's begin with the first question. Question 1. Messenger RNA and transfer RNA are involved in protein synthesis. The diagram shows the sequence of bases in part of a DNA template, which is the antisense strand, the messenger RNA molecule formed during protein synthesis. So here we have the DNA template strand, and we were asked to complete the diagram to show the sequence of bases in this messenger RNA. If DNA has adenine, messenger RNA is going to have uracil because messenger RNA will not have thymine. So if we have thymine here, it's adenine, guanine, cytosine, cytosine, guanine, cytosine, guanine, guanine, cytosine, thymine, adenine, and thymine, adenine. So that should be the base sequence. Next, they say name this stage of protein synthesis. Of course, this is DNA transcription. And name the enzyme used in this stage of protein synthesis. The enzyme is RNA polymerase because we are trying to transcribe DNA into messenger RNA. In part B, the diagram shows part of a transfer RNA molecule. They say compare and contrast the structure of messenger RNA with that of transfer RNA. Questions about compare and contrasting are looking for similarities as well as differences. And in this case, we're interested in looking at the structure. So the similarities are both as single-stranded because they're both RNA and both have adenine, cytosine, guanine, and uracil. They do not have thymine. In place of thymine, there is uracil. Now the differences are messenger RNA contains the codon, while transfer RNA contains the anticodon. Messenger RNA is not folded, so it has no hydrogen bonds. Transfer RNA has foldings with hydrogen bonds between them. And then transfer RNA has an amino acid holding site, but messenger RNA does not have. So this brings us to the end of question 1. Let's continue to question 2. In question 2, the graphs show how the pressure and volume of blood in the left ventricle change during the cardiac cycle. So the first graph shows us the pressure in the left ventricle, and on the horizontal axis we have time in seconds. So we see the increase and decrease in blood pressure as time passes. And for volume, there is a decrease, followed by an increase, and then another decrease as time passes. Here they say, which letter indicates when the atrioventricular valves close. The atrioventricular valves close before the ventricles begin to contract. So contraction of the ventricles is going to increase the ventricular pressure. Point R is when the volume is beginning to increase, so that is going to be my answer. Next, they say calculate the time for one cardiac cycle. I chose from this point here to the next corresponding point here. You could choose any point provided you can get it properly. So, in my measurements, I think you can see it was about 0.73 seconds. The total time from this point here to that point here, it was 0.73 seconds. In part C, the volume of blood in the left ventricle changes during the cardiac cycle. Explain these changes. So I say during atriosystole, this is when the atria are contracting. Blood is going to pass through the atrioventricular valves, and the volume of the left ventricles is going to increase. And again, remember, we're interested in the changes of the ventricle volume or ventricular volume. Now, when this volume increases, then we go to ventricular systole. That is when the ventricles are contracting. During ventricular systole, blood passes through the aortic valve, so ventricular volume is going to decrease. And finally, during cardiac diastole, there is some increase in ventricular volume as the ventricles start to fill. So this brings us to the end of question 2. Let's continue to question 3. Question 3. Sucrose is a disaccharide made from glucose and fructose. Glucose is joined to fructose by 1,2 glycosidic bonds. The diagram shows a molecule of glucose and a molecule of fructose. The carbon 1 on glucose and the carbon 2 on fructose are numbered. They want you to complete the diagram to show a glycosidic bond between the glucose molecule and the fructose molecule. Because this is a 1,2 glycosidic bond, it's going to be between carbon 1 of one molecule and carbon 2 of the other. So a glycosidic bond includes an oxygen bonded to the carbons on the other side. So this is what I showed. Next, they say complete the formula equation for the reaction that makes sucrose from glucose and fructose. We know that combining these two involves a condensation reaction where one water molecule is going to be lost. So the number of carbons are not changed. So 6 plus 6 gives us 12. So it's going to be carbon 12. The hydrogens are going to be added up, but they lose 2. So 12 plus 12 is 24. But when you take away the 2 in water, it leaves 22. So hydrogens are 22. and the oxygens, we add them, they are 12, but minus 1 that goes to water leaves 11. So this is my formula. In part B, the concentration of sucrose in a solution can be determined by a test. The chart shows the steps involved in this test. First is break sucrose down into glucose and fructose using an enzyme, and then add a chemical that binds to fructose and changes color. So this chemical is only going to be binding to fructose, and then you measure the color intensity of the solution formed. So since we're measuring color intensity, it means the more fructose formed from the digestion, the more the color intensifies, and therefore the intensity is going to be greater. They say the color intensity is directly proportional to the concentration of sucrose. The graph shows this relationship. So from this graph, we see the concentration of sucrose. As it increases, we see the color intensity increasing. And again, this is because if we have more sucrose, more fructose is going to be produced. The more the chemical binds onto the produced fructose, the more the intensity of the color observed. Down here, they say give a reason why the color intensity is directly proportional to the concentration of sucrose. So here you say because as the concentration of sucrose increases, more fructose is produced, so the color intensity increases. However, a direct relationship is because each sucrose molecule contains only one fructose. That's why we see it increases by the same magnitude. Next, they say suggest why the line does not start at the origin of the graph. So the origin of the graph would be at the point here, but it starts at a point which is around here. This could be because if we have very low sucrose concentration, yes, there will be some digestion, but the fructose produced is going to be so small to cause a significant color intensity. So I said because color intensity will be too low. The next I say state why the concentration of maltose and lactose cannot be measured using this test. This is because maltose contains only glucose, so there is no fructose to be detected, and lactose contains glucose and galactose, and again, those are not fructose to be detected, so both galactose and glucose can bind to the chemical, so there will be no detection in color intensity. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Hydra are small animals that live in water. They have a cylindrical body. The drawing shows a hydra. So this is the hydra. The hydra has a body length of 1.8 and a diameter of 1 millimeter, which is the magnification of this drawing. We know magnification is going to be image size divided by the actual size. So my image size was 48 millimeters, and if I divide it by the 1.8 millimeters, I got 26.6667, which is approximately 2.67 times 10 power 1, and D was my answer. Next, they say which is the surface area in millimeters squared of this hydra. They want you to use the formula surface area is equal to 2 pi RL. Of course, radius is going to be R and L is going to be the length. And since we're given the diameter, it means the radius is going to be half of that, which is 0.5. So our surface area is going to be 2 times pi times the radius times the length which we have, and that gave me 1.8 pi. That's my final answer, which is B. Next, they say another hydra has a surface area of 2.3 pi millimeters squared, a body length of 1.8 millimeters, and a diameter of 1.2 millimeters. They want you to calculate the surface area to volume ratio of this hydra. And again, they want you to use this formula. Volume is pi r squared L. So here volume will be pi into the radius is going to be half of the diameter, which is 1.2 divided by 2. So you square that times the length, which is 1.8. And I got 0.648 pi. That was my volume. So I got 3.55. Actually, it was 3.549, so I rounded off to 3.55. In part B, a hydra has a hollow body that is made up of 2 layers of cells. Explain how a hydra is adapted for gaseous exchange. Here they say the body is hollow and only 2 layers of cells, so this means there is a thin distance for substances to come into the hydra, so the fusion is going to be efficient to supply the things required within the hydra. So I said it has a large surface area to volume ratio, so oxygen present in the water enters the hydra by diffusion. Because the body is made up of 2 layers of cells, the diffusion distance is shorter. So this brings us to the end of question 4. Let's continue to question 5. In question 5, stents are used in the treatment of atherosclerosis. Stents widen the diseased coronary artery so the blood can flow through to the heart muscle. The diagram shows a stent. So this is a stent inserted into the coronary artery. They say stents can damage the endothelial cells lining the artery and trigger the formation of blood clots. Patients who have a stent fitted are given anticoagulants. Explain how damage to the endothelial cells results in the formation of thrombin. Thrombin is an enzyme that is involved in the blood clotting process. So that means if the damage to the endothelial layers occurs, it's going to trigger the release of thromboplastin from the platelets. So I say damage to the endothelial layers triggers the release of thromboplastin from the platelets. Then thromboplastin is going to convert prothrombin, which is inactive, into active thrombin, which is the enzyme. In part B, they say give one risk of treating patients with anticoagulants. Remember, anticoagulants prevent the clotting of blood or formation of clots, and that means there is a chance that the patient could bleed without stopping or internal bleeding could occur. So I say taking anticoagulants affects the blood clotting process, so the person can bleed internally or bleed excessively if injured. Next, they say the photograph shows a tropical pond tick. This pond tick produces a peptide called vireogen. Vireogen binds to the active sites of thrombin and prevents the blood clotting process or prevents blood clotting. Remember, if it binds to the site of thrombin, it means fibrinogen is not going to be converted into fibrin. They say vireogen is a peptide consisting of 32 amino acids, which is the minimum number of bases in the gene that codes for this peptide. 32 times 3 because each amino acid is coded for by one full codon, so this is going to be 32 times 3, giving us 96, plus the stop codon, so 96 plus 3 gives us 99, so my answer was D. Next, they say explain how vireogen prevents blood clotting. Like I've already said before, it binds to the active sites of thrombin, preventing fibrinogen from binding there, so the fibrinogen can be converted into fibrin, and without fibrin, blood cells can be trapped to form the clot. Here they say thrombin slowly splits vireogen into two molecules. Explain how thrombin splits vireogen. It means vireogen is going to bind into the active sites of thrombin, and then this is going to lower the activation energy for the reaction, meaning that reaction will occur at lower temperature. And then a peptide bond within the vireogen is going to be broken by the hydrolysis reaction to form two molecules. The molecules then leave the active site of thrombin. Next, they say the effect of vireogen on the mass of blood clots forming inside a stent was investigated. The masses were compared with another anticoagulant, which is drug H, and the graph shows the results of this investigation. Each plotted point represents the mass of blood clot in one stent. So from our graph, we see the results for vireogen and the results for drug H and the corresponding masses. We can see these masses are spread over a wider range. Some are lower, some are more, and we see here those for vireogen giving almost the same mass. So they say comment on the results of this investigation. So from these results, we can see there is more mass with drug H in comparison to vireogen. So I say generally drug H resulted in more mass of the blood clot than vireogen. The blood clot masses with drug H had greater variation than with vireogen. They are wide spread out. I already say there is a lot of variation in the masses. There is no mean mass or error bars, so the results are not valid. And the graph does not show the data for a control. And again, here they say comment. Wherever they say comment, you have to talk about the validity of the data. So no mass of blood clot without any drug, so a conclusion on effectiveness cannot be made. Because they would have showed without a drug what the masses are, and then we can compare the masses without the drugs and then the masses with these two drugs in order to make a fair conclusion. So without that, we cannot conclude on the effectiveness of the drug. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Insects and other invertebrates are a good source of protein, unsaturated fatty acids, iron, and other nutrients. Recent research has shown that some insects and other invertebrates have a high antioxidant content. How many of the following statements are true for proteins? Number 1, proteins contain only the elements carbon, hydrogen, and nitrogen. That is wrong. Proteins can be made of more than one polypeptide chain. That's true. And proteins contain hydrogen bonds. That is also true. And again, to emphasize on this point here, I wrote some examples. Hemoglobin, which has 4 chains, and insulin that has 2 chains. Here, of course, proteins contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. So there are only 2 true statements, and my answer was C. Here they say which row of the table is correct for an unsaturated fatty acid. So the first part says contain carbon-carbon double bonds, that's true. And then the carbon to hydrogen ratio compared with a saturated fatty acid. Here I gave an example. If they're unsaturated, they will have a carbon-carbon double bond. I gave an example of this. And if they're saturated, there is no carbon-carbon double bond. And again, these examples are not necessarily fatty acids, but I used hydrocarbons to show you quite easily. In this compound here, we have 2 to 6, which is going to be 1 to 3. We can see this is less than that. So it means the carbon to hydrogen ratio is going to be higher in unsaturated fatty acids. So my answer is going to be a D. In part C, they say name one organic molecule that contains iron. That's easy, it's hemoglobin. Part D, explain why dietary antioxidants are important. The antioxidants reduce free radicals, and this is going to reduce the damage to the endothelial cell layer in the blood vessels. This prevents or decreases plaque formation, and therefore the risk of atherosclerosis, cardiovascular disease, and stroke is going to be reduced. In part E, the antioxidant activity of some insects and arachnids was investigated. The table shows the dietary habits of organisms used in this investigation. So we can see the first 3 here eat animals, and then the next one eat plants. From the graph, they say the graph shows the main antioxidant activity of extracts made from these organisms. So again, we can see the data here, but I labeled here. These are the ones that ate animals from the table, and these were the plant eaters. We see overall that plant eaters have more mean antioxidant activity in comparison to the animal eaters. We see the data for animal eaters does not have any overlap, but some overlap exists here. The antioxidant activity of orange juice is 0.8 AU per 100 grams. Calculate how many times more antioxidant activity there is in 100 grams of grasshopper extract and give your answer to 2 significant figures. Let's go to the grasshopper extract. Here, this is the grasshopper extract, and we see the mean is 2.3 AU. So this is what I got from the graph. So 2.3 divided by 0.8, which we have here, is going to be 2.875, which is approximately 2.9 times more, and that was my answer. Next, calculate the percentage difference between the antioxidant activity in black scorpions and in crickets. So again, we go back. The mean for black scorpions is about 0.1, and the mean for crickets is about 1.9. So the difference is going to be 1.9 minus 0.1 divided by 1.9 times 100, or you could say 1.9 minus 0.1 divided by 0.1 times 100. This gave me 94.7%, and this is 1800%. So both answers are acceptable because they ask for percentage difference. Then they say comment on the results of this investigation. They want you to use the information from the table and the graph to support your answer. From the graph, because different organisms had different mean antioxidant activity, those different organisms had different levels of antioxidants. The difference could be attributed to their diet. Those that feed on plants had greater mean antioxidant activity than those feeding on animals. They included error bars that shows the results were repeated. However, the error bars were bigger in plant eaters than animal eaters, showing greater variation in plant eating organisms than animal eaters. And that means there were different compounds in the different plants that were consumed. So this brings us to the end of question 6. Let's continue to question 7. Question 7, prenatal testing is used to screen for genetic disorders in developing embryos. Phenylketonuria, PKU, is one genetic disorder that is screened for by prenatal testing. This disorder is inherited in a similar way to cystic fibrosis. Explain why a healthy couple might choose to have a prenatal test for PKU. They want you to draw a genetic diagram in your answer. So here I began with parental genotype, and I expressed them as heterozygous. The parental gametes are going to be A as well as a small a, where this is the recessive one and this is the dominant one. So I drew a genetic diagram here to show you the possible gametes as well as the possible offsprings that can be produced. And we see 3 offsprings are going to be okay. The phenotype will not show any disease, but at least one is going to show some disease. So the offspring genotype is going to be AA to AA2, that AA is 1 to 2 and the offspring phenotype is going to be 3 normal and 1 with a disorder. So I say down here, parents might want to test to prevent having a child with a disorder. Both healthy parents could be heterozygous for the disorder, and as shown above in the diagram, 1 in 4 offsprings has the probability of becoming homozygous recessive. and those who are homozygous recessive are going to show the disease. In Part B, a group of women who had recently given birth to healthy babies were asked about their opinion on screening for 4 different genetic disorders. There is A, B, C, and D. They were asked 3 questions about each disorder. Would they be screened themselves with a blood test? Would they have amniocentesis to screen for the genetic disorders? Would they have a pregnancy termination or abortion if an unborn child was found to have a genetic disorder? So the graph shows the percentage of women who said yes to each question for each disorder. For disorder A, we see more said blood test. Actually, for all disorders, we can see more mothers or more women were willing to take a blood test. And then the next was amniocentesis, and lastly was for termination. For each disorder, fewer mothers were willing to do termination. When we go down here, there is a table. They say the table gives some information about each of these genetic disorders. So the genetic disorders we see here for A, it causes muscles to be weak and movement is difficult. Symptoms can be managed. Some types are lethal in childhood. So if they are lethal, some parents could be scared and some could opt for termination. This is treatable and does not shorten life expectancy. And with C, it's treatable and most individuals live into adulthood. For D, there is intellectual disability. Medical care results in individuals living healthy lives. They say discuss the responses to the 3 questions. And again, in my discussion, I'm going to read what I wrote, but of course, I already talked about some of them. The percentage of women opting for blood tests were close and the highest in all disorders. This would be a less invasive method with less negative effects on the unborn child, but this does not consider paternal DNA. The percentage of women opting for amniocentesis was second highest, almost the same for all disorders. This would be because they were eager to find out about the genetics of their unborn child to make adequate preparation. However, amniocentesis has higher chances of miscarriage. And then for all disorders, fewer women opted for termination. This could be due to ethical reasons. However, the percentage to terminate was high in disorder A. This could be because some types of disorder A are lethal, so the mothers want to save their unborn child or children from pain. So this brings us to the end of question 7. Let's continue to question 8. In question 8, the risk of developing cardiovascular disease is influenced by a number of factors. Risk assessment calculators allow a person to calculate their own risk of developing CVD. The person completes a questionnaire about their health and lifestyle. This information is then used to calculate the risk of the person developing CVD within the next 10 years. The table shows the information required by 2 different risk assessment calculators. There is RAC1 and RAC2. So the information given here for age, we can see for both, yes, they take age. Blood pressure, of course, both test for that. Smoking, only method 1, method 2 does not. And I think this is going to be bad because smoking is a risk factor for CVD. Total cholesterol, both of them take it. HDL cholesterol, only method 2, method 1 does not. But also this is a problem because HDL cholesterol, or the ratio of HDL to LDL, is important in determining the risk of CVD. Now for age, this is carried out because as people age, the risk increases. For blood pressure, the higher the blood pressure, the higher the chances of damaging the endothelial cell lining, the higher the chances of forming plaques. And then total cholesterol, the higher it is, the more the chances of plaque formation because it can be deposited in areas where the endothelial layer has been damaged. So they ask how many of the risk factors listed in the table are not influenced by lifestyle. The only non-lifestyle factor is age, so I wrote 1. The others are all lifestyle factors. So they say explain the effectiveness of these 2 calculators in the assessment of risk and whether other factors should be considered. So for age, as people get older, the risk of CVD also increases. And then for blood pressure, high blood pressure damages the endothelial cells, and this leads to inflammatory response and consequently plaque formation. For smoking, smoking increases the risk of CVD because smoke contains particles that irritate the endothelial layer in blood vessels, and this can result in atheroma formation, which in the end leads to CVD. For cholesterol, or total cholesterol, the more cholesterol consumed, the more cholesterol builds up in plaque within the blood vessels. Now for HDL, knowing the amount of HDL enables calculation of the HDL to LDL ratio, which can be indicative of the risk of CVD. Now, in the calculators, other factors were not included, for example, genetics, gender, salt intake, diabetes, activity level, stress, the quantity or number of cigarettes consumed by one person per time. These were all not included in both methods. And we know for genetics, some people are genetically predisposed to have CVD. Those who consume high amounts of salt, this leads to blood pressure, and lack of activity leads to an unhealthy heart. From the study, both methods of evaluation are missing crucial information in determining the risk of CVD. Method 1 is missing HDL cholesterol, and method 2 is missing smoking. To determine which method is more effective is hard since people may not know their HDL total cholesterol while answering. In part C, the calculators work out the risk of a person developing CVD in the next 10 years, explain why the information entered by a person may lead to an understatement of their 10-year risk of CVD. This is because some risk factors like genetics, numbers of cigarettes may not have been included during the calculation, and some people may not be honest on the test. Next, they suggest why these risk calculators may not be suitable for everybody to use. This is because a person may already have a high risk condition. Some people are already obese, they already have diabetes, and some are genetically predisposed to this. And some people may not know all this information about themselves. Like I said already, they may not know their total cholesterol, they may not know the amount of HDL, and so on. Next, they suggest why these risk calculators estimate the 10-year risk of developing CVD. This is because CVD does not develop in one day. It takes time because lifestyle factors contribute. So I said because CVD takes time to develop depending on the person's lifestyle and age. So this brings us to the end of question 8. Let's continue to question 9. Question 9, some marine animals are osmoconformers and some are osmoregulators. Strict osmoconformers have a cell solid concentration that matches the solid concentration of the seawater. Partial osmoconformers have a cell solid concentration that varies with the solid concentration of the sea. Osmoregulators control the solid concentration of their cells when the solid concentration of the sea changes. The graph shows the solid concentration of the cells of 3 marine animals. We have P, Q, and R. On the graph, we see solid concentration of the sea as it increases. For some, we see it remains the same. So these are osmoregulators because it doesn't change. But we can see the others are strict and partial because it's going to change. This one here changes as the sea changes, and they have a linear relationship. This one here has some control, but then it changes as the sea concentration changes after the concentration of the sea water increases. So they say which row in the table identifies the marine animals P, Q, and R. P is going to be partial because you can see it has the ability to a little bit control in the beginning, but as the concentration increases, it cannot control that much. And then we see Q is going to be a strict osmoconformer because its concentration changes as the sea salt concentration changes because they have a linear relationship from the beginning. And then R is going to be an osmoregulator because it ensures that its salt concentration remains the same no matter how the solid concentration of the seawater changes. So they say explain the advantages for osmoconformers of having cells with solid concentration similar to that of the seawater they live in. So if the concentration is the same, there is no need to regulate that much. So this ensures that less osmosis occurs, so they will not excessively lose water or dry out. Also, they will not excessively gain water for the cells to swell and burst. So in simple terms, less regulation is going to occur. In part B, the table shows the concentration of ions in seawater and in some osmoconformers. So we see the seawater concentration and then that in the cells of osmoconformers. We see they have different concentrations of the different ions. So the first question said estimate the chloride concentration of the cells in jellyfish. My estimation was 560, but 550 to 580 is acceptable. Next, they say calculate the percentage difference in the concentration of magnesium ions in the lobster compared with the sea urchin. And they want you to give your answer to 2 decimal places. We go back to the table. We can see there is this concentration and that. So the difference is going to be 54 minus 9. So 54 minus 9 gives us 45. And therefore, percentage difference is going to be 45 divided by 54 times 100, or 45 divided by 9 times 100. And again, this is percentage difference. So the answer is 83.3% or 500%. Next, they say these 3 animals are osmoconformers even though they have different concentrations of each ion in their cells. explain why these three animals can be described as osmoconformers. No matter the different concentrations of the ions they possess, the water potential within the cell is determined by total ion concentration, not just the individuals, but the total. So I said water potential is affected by total ion concentration and not the individual types. So since they have the same total ion concentration, similar to that of seawater they live in, they have the same water potential as the seawater. Then suggest why osmoconformers do not have the same concentration of each ions in their cells. This is because there is a possibility that some ions are needed more in specific cells, and also these osmoconformers could be feeding on different diets that can deliver different ion concentrations. So I said ion concentration can be affected by their diet as well as their location in the sea. They could also absorb different ions based on their cellular requirements, and that is going to cause the different concentrations within the cells. Lastly, they say energy is needed to maintain the concentration of ions inside the osmoconformers. Explain what this energy is being used for. Energy is used, of course, for active transport, so that ions are moved from the sea into the organism against concentration gradient using ATP, and that is going to retain the ion concentration to the required level within the cells. So this brings us to the end of question 9 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.